coming, everybody. I want to introduce Ángel Robles Fernández here. You've probably seen him around. He's a postdoc, sits over in Noguchi, down at the end of that corridor with all the cubes in it, one of the offices down there. He did a master's in physics from the University of Veracruz, and then went and did a PhD in ecology and evolution at Arizona State University. So he has both quantitative and skills and kind of ecological wisdom. Before that, he did machine learning in the steel industry as a sort of data scientist. And now he's here. He's been here for a year or so. He's going to be here for a couple more years. I'm sure he'd want to talk to you if you haven't already gotten to know him. So introduce Ángel. Thank you, Ángel.
Thank you, Dan.
You hit the lights. Hit the cord. Thanks, guys.
So I will present what I've done in the PhD program. So just a disclaimer, this is not the typical ABC research project that we have in the PhD, you know, interconnected papers. This is a set of 3 independent research projects that are around some ideas in between, but not exactly related with each one with the other. So I will try to summarize as fast as I can. But the main goal of the PhD project was to create a system that could potentially predict new host-parasite interactions. So I will start about this. So this talk is, as I said before, I divide my dissertation in 3 chapters, and I will present the main ideas of each chapter and the ongoing projects I have. But how are related is not straightforward. So basically, what I do is create software tools in the first and the third chapter. And in the second chapter, I develop a theoretical framework and then I test it. This theoretical framework is how we relate the concepts we have in the niche theory with population genetics concepts. I developed that myself, and then we apply with target species. So that chapter is a little different to the 2, because 1 and 3rd chapter are software development to solve a specific task. That's why this structure is very strange, but it's my dissertation. I will try to be the most concise I can, okay? So, which is the problem we have in general? We decided to study the system because, as you know, we have this COVID-19 emergency, and I was in the middle of the emergency doing my research in the master's in physics, and in between I was learning how to apply machine learning algorithms in ecological studies. So the problem suddenly reached me. It was something that I was not planning in that moment. I was planning with my master advisor how to apply machine learning in ecology and evolutionary problems. But in that moment, COVID-19 starts. So we get confined, and we see that was a good opportunity to do research specifically in that problem, because it was something that just appears. And then, you know, we study that because most of the pandemic issues comes from our interaction with the disease confined in the animal species in the nature. So this is a zoonotic process, and 60% of the emerging infectious disease come from that interaction. So we want to know how to potentially model that. And this is because, as you remember, this creates a big problem in the world. So it's a very important thing to do. So that was, then after the master's, I get invited to the PhD, and the main goal was to create a kind of machine learning predictive framework to deal with these problems in the future or the near future, and hopefully have software tools that can help in other near future pandemics, potentially we can have. So that's the main goals. And we split the project in 3 branches. And what's just the first chapter is how we can automate the extraction of the data or create tools that help us to generate new information from the data we have, or how we can create enough new knowledge up with the previous knowledge we have. So we try to create software to put in the pipeline that automatically discriminate the information that can be useful or not. That's the first chapter. Then the second chapter was something that I developed in the middle and was not exactly related with the pandemic issue, but more with the ecological and evolutionary process behind the interactions. But before to reach the interaction thing, I go deeper and more in the base of the nature of the relationship between genetic diversity and the concepts of niche. So it's more different to the other chapters. And then the third chapter is a predictive framework by itself that potentially has the ability to find new interactions between host and parasite that we have never seen before with the knowledge we have now. So I put these 3 branches of my research in 3 research products, and I just give some names because it's important to have these projects with their own name, okay? So basically that was my research strategy. Chapter 1, machine learning that can classify and try to automate the process of the data extraction. The second is a framework that relates genetic diversity with the concepts of the ecological niche. And the third is the machine learning that tries to predict these interactions. So I will talk about the chapter 1. Basically, I don't have enough time to develop the full system as I was designed, but what I do is just a system that tries to classify the literature. When you have these kinds of pandemic issues, what you want to do is to have a tool that gives you the papers that you need more in a specific area. I mean, in this case, the pandemic, but could be any area. So what you want is to have something that, given some words, gives you the papers, retrieve the papers from the database and gives you the papers that are more accurate with what you need. That's the nature of this tool. So I put this to the name of LAX because, you know, with LAX, there are gaps in the information. When you go to the NIH database, the PubMed database, and you search something, you don't know how it's working the ranking behind. You just use it. And that's not always is good because the ranking the PhD, the NIH is using is not clear how it works. So what I designed here is a system that training with the specific papers of the area you want, retrieves the papers you need in the ranking you know that is closer to the information you provide to the system. And that's the idea. So this was designed, the implementation of this was designed for synthesize the host parasite data, but you can actually train in any area you want. Okay, so the main idea, how it works is, this is not a fancy language model, you know, a large language model that nowadays we have with ChatGPT or the other language systems. This is a cheaper version of that. Instead, we have, I mean, because I don't have the resources to train a large model, the large language model, what I do is just to compare the information in the papers I provide using a very fast algorithm, but that actually works for the purpose you want, that is classify the text. So automatically process the words and find which other papers are close to this word space. Imagine that you have words in a bag and you look for the paper you want and see how close are in the bag you have. But we have the problem that we only have one class of paper, the papers I want, the positive class. I don't have the negative class because I don't have time to curate which are the negative papers. So I use a machine learning approach that is called positive and unlabeled. That it means that I just provide the positive cases and I take random papers to train as unlabeled, that's unknown papers, and then the machine just select which are the closest of the papers I provide. And then we create a small app to test it, and basically connects to the NIH PubMed database and train it as I give the papers, returns the papers that match better with the words I want to fit, but in my own rank. And then I compare this with a tool that NIH developed for the same purpose, but they do with some similarities, but it's not exactly the same. So that's the methodology. Basically, we have these papers, I chunk in words, I put some similarity metrics between them, and then you go to the website and just type what you want, and it retracts you the papers that are closer to that. That's it. So I measure the performance of my system compared with the performance of the NIH system. The system they are using is this literature suggests. It suggests do the same. You provide some papers, automatically it trains a model, and then gives you the papers in the PubMed database that are closer to the papers you provide to the system, and that's it. And gives you a metric how it classifies. So I validate this with an external database to compare the performance of PubMed it suggests to my system, and that's it. So this is the system NIH has implemented. You can go there and use it. I mean, it's not bad, but it's you cannot use it as programmatically in a programmatically way because it's just a web interface and it's just designed for that. It's for medium. It's not for advanced users. It's just for users that don't want to do more with these things. And it's okay from my point of view. So you provide papers, it trains a model, and then classify the papers in the PubMed dataset and gives you the rank of the papers, and you can grab it. The many do the same. I have some comparisons here, but basically the point is that I develop an R package that do this, and you can modify the package as you want. So instead of train with the specific papers in PubMed, you can train it with the papers you want in the area you want, and that creates more flexibility. So it's not designed just for medical purpose like PubMed that has a specific literature in the database. You can use this in any datasets you want. Okay, and this is the app I implement there. So you can test it if you want, and you search the term you want and returns back the papers ranked in the similarity of the information you trained. In this case, we trained for host-parasite interactions, and these recommend you papers related with the search term you are finding. And that's it. And this is their patch to do the same by yourself. So these are my results. As you can see, I have very similar results comparing with the lead suggest, but in my case, the true positive rate is higher in the validation dataset. That means that in the training process, the lead suggest software tends to over predict and can give a lower score when you present new data to the system. That's why it's important, the validation process, because this is new data that has not been used before the train and the test process, and my system responds better. That's why I show this. And this is measured with this overfitting gap. The overfitting is that you feeding with the knowledge you provide to the machine, and the machine cannot predict new information. So my system relies in different bases than the NIH system, and it's able to have a lower overfitting gap. So it has better performance in new scenarios. That's it. And this is the conclusion. We can overcome the limitations we have sometimes thinking more how to implement these things. And my goal is to make these recommendation systems more accessible to first users that doesn't know how to implement a model like these language models. My goal is not to compete with ChatGPT or no, my goal is more direct and a pedagogical way to use this as a learning process in machine learning. And then I also can use it for my own research in the middle of that. Okay, that's the chapter 1, a system that classifies the literature from the NIH. Second chapter, this is completely different. The main idea is how we can correlate, sorry for my English. How can I relate the structure of the genetic diversity between the populations and the structure of the ecological niche? So I will be fast in this, and if you have any question at the end, I can try to answer. But the full development of the theoretical framework is on the paper, and the main goal here is not to put lots of formulas there. It's just to explain the result and how we get there. Okay. So what we do basically is model environmental niche with the assumption that we neglect the interactions, just for this case. You can do that if you use species that have the same distribution as the interactions that the species can have potentially. For example, if you model some species that live in some specific pine trees, the distribution of the pine trees and the distribution of the species should be similar, so we can neglect the biotic interactions. And we just use that information to try to understand which was the past distribution of the species that is using environmental information to the past, from the present to the past. And then we try to infer what happened with the structure of the populations, the genetic structure of the populations in the sites where the environment remains the same along all this time. So for the purpose of this project, I have some limitations and I take some cases, but the idea behind is that. So I have 2 questions in this paper. If the sites that remain stable in this environmental niche remain the same along the time, these sites correspond to the historical endemic area. That's it, the area where the species can live, even if the environment changed, the species remain the same area. That's the first question. And the second, if the genetic structure of the populations is related with the sites that are environmentally stable along the time. So those are my 2 research questions. And that's why I developed these 2 objects. For the first, that is the limit these endemic areas, historical areas where the species can be suitable a long time. I call the SPA. This is the suitability prevalence area. That's the portion of the environmental space that that let the species be there, but remains in the geography along all this time. And then we test that the genetic structure is related with this SPA, this suitability prevalence area, to explain the genetic structure of that species. In this case, we take this squirrel as a model to test this theoretical hypothesis, because checks with our goals that overlaps with the distribution of the host of the squirrel that are specific pine trees in Mexico and United States. So it's a system that you can imagine as an island, because it's a pine forest surrounded by desert or other habitats. So these are our objectives more detailed. We model the ecological ditch model of this squirrel, Cirrus abertis, and we project to the vast environmental information. In the resolution, in that moment, I have access. Now I can do better, but in that moment, I have 120,000 years of information in 2,000 slices. So I have 60 slices of time to see what happened with the distribution of the species, and then find this region that I found the SPA that is the superposition of the information and remains along all these years. And then I try to explain the genetic diversity through this SPA using a model I developed, and expect that the sites where the environmental conditions remain along the time let the species create a more genetic diversity, populations with higher genetic diversity. If you compare with the sites where the species is not suitable all this time. That's the main idea. So I will explain with more detail this part. I'm using a measure for this study that is called the fixation index. This is the proportion of the genetic differentiation you have regarding with the total of all the genetic diversity in all the species. So you can compare populations and gives you an idea how they differ. Suppose you have two populations. If they can get in contact and they have inflow and have enough population size, the fixation index will be zero, because you can imagine it's like which portion of this can share alleles with this. So because all the portion can be shared the same, so this index is zero. So when this index is zero, it means that the populations are genetically similar. And in the second case, suppose you draw a line here. So when there is a barrier, these populations diverge a long time, because there is no gene flow and the population starts to get differences a long time, and in some point could be completely different. So this is a mechanism that probably some species can explain how two species can split in some moment. So when they are completely differentiated, there's no gene flow between there is no other acting other forces. They have a very high FST. These range from 0 to 1. So when they reach 1, they don't share anything in between them, and they cannot be considered the same species even. Okay, so in the population genetics theory, you have a balance of evolutionary forces. I will just summarize this, but the main idea is that you have some of these push the fixation index up, like the genetic drift, because if you have small amount of population, that increase the change in the random alleles that are there. That creates a differentiation higher than when you have larger populations. The opposite is gene flow. When there's gene flow, you have new use of genes in the other population, and this tends to go down. So you have these two forces cooling. And the mutation is something that in this case we neglect, because this is slightly increase the genetic diversity, but for the time we have in this example, we can neglect it. And that's not true for all the species, but for our model, we neglect the mutation, and we have this famous equation that is the right island, the island model of rights for the fixation index. Then I start to think about this and how we can relate with the environmental suitability along time. So this is the main hypothesis we have. If you have environmental conditions that remains all the time, you can expect that the populations are stable there, and that creates the balance of the forces are enough to sustain the increasing of the genetic diversity because the population is stable, because the environmental conditions remain stable. So as this suitability remains stable along the time, the fixation index tends to go to zero. But if you have the opposite, that you have some cases where the suitability can go down and up and down and up because not all the time there are conditions there, the fixation index tends to go up because you can have potential extinctions, you can have barriers that limit the migration. So this is the main model. I have the full development of this equation in the paper, but it's basically it's relating the suitability prevalence a long time with the effective population size here. That's it. That's the trick. With that in mind, suppose, for example, you have two populations here. So one of these is this red one, are inside what we consider in this case the niche all the time. So it remains stable along these 120 years. And you can imagine that the fixation index, if you measure these two populations, will be lower here if you compare with the other populations. And the blue one, the P1, it's in some places that now you can see that the species is there, but you can think this that the conditions will not remain along all these 100,000 years. So we see that now is here, that inside the niche, but could be outside the niche in some moment. So that creates extinctions or barriers like this. This is a Sonoran desert. So if there is no connection between them, there's no flux of genes. So this goes up and this goes down. And should be that this is true. So let's perform all the calculations to see if that happens. So this is the methodology we use. We fit a niche for the zero severity and we project to the past in these time slices. Then we gather the information with this suitability prevalence area. And then we feed the models because we have a number of populations. So we feed one population with respect to the others, a linear model, then the second, a linear model, the third, a linear model. And what we expect is the populations that are compared with the population that remains stable, we have significantly explained this process and the other doesn't explain anything. So that's what we want to know. This is the original data I use. I don't compute it. I just read and grab something that it works for my theory. In this case, I use this data from Bono et al. They calculate the fixation index along 9 populations, and it's curious because this one, it's here. This is divided by this barrier that is the Colorado River. This is the Great Canyon, and this is the big desert. So it's very difficult for the species reach here now, but could be that in some moments there's a bridge that could be connect these, the same here. So this region is known as the Sky Islands. So this is a perfect model for me. And I feed the niche for this species This is the actual area of the species in my model. Then I project to the past. So I can see the expansions and the contractions of the area. And I see a peak here that is related with the maximum last glacial maximum, where all the ice sheet goes down, and that's create a push in the system that creates connections between the pine trees, and the species goes down, 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 and reach this region, and everything is connected here in the peak. But if you see, there are some moments that the species is near to the relative area of the species in my models are very scarce, because the conditions are not remain everywhere and just remain in some very specific places. So we stack all this information to find which places the conditions remains along this time. Remember, we call this SPA, the suitability prevalence area. And we have this map in the contour. We have the black is the 90% of the time the species remains with conditions. The blue is 50% of the time. You see here, here, and here. And then we track the population genetic information and perform our logistic regression for all the population. So we have 9 logistic regressions. Sorry, not logistic regressions, no, sorry. Linear regressions. These are linear regressions, sorry. And we have 9 of them, and it fits good. That makes our theory, it's good. And then we use the only model that has statistical significance, and plot back to the geography to see how the fixation index change along the geography. So that's the final output, and this is the paper we published with this information. So these are the results of all the 9 linear models. As you can see, the only statistical significant model is this population that correspond to this one. So our suitability prevalence area shows that the conditions remain stable there. This is the Sierra Madre, the West Sierra Madre. And it seems like from this point of view, the structure of the genetic population shows that some of these patch appears and disappears, and now they can be suitable, but not all the time. And that's the summarization of this. We have a high explanatory power in our model, but it works under all these assumptions. So I think this is not fully generalizable to other systems, but with some caveats, it works. And that's it. The future work is to test with different species that doesn't have these young distributions, explore how to add the information with the gene flow in a more independent way that I suppose in just if the suitability remains between the 2 populations, that connects the genes, but not exactly how it's happening. And then testing with many ecosystems and other places in the world. Okay, I will try to do my best in the third, because I don't know how much time I have. But okay, the third chapter is completely different to the other 2. This is a predictive framework that aims to find new interactions that we have never seen before with the knowledge of the information we have now. Okay, so basically, I do some search in the internet, and before the pandemics, it's obvious there are few teams that was looking for how to predict host-parasite interactions with machine learning. Actually, I just find 4 papers, 5 in 2019, 4 in 2020, and then boom, in 2021, 7 papers, and so on. Now it should be more. So basically, in that moment, there was 3 main ways that the people try to predict these host-parasite interactions. Using graph theory and neutral analysis, that was some groups doing that. Others consider the phylogenetic signal as a determinant. That means that if you have two species that are phylogenetically close, they should share the same parasites. That's an old idea from some researches. And they add the topology of the network to improve that. And the third is consider either traits of the parasites on the host and try to predict the links. That's it. Summarizing, if you have traits similar in the host and traits similar in the parasite, they match. So they analyze these trait matrices between both and see which match. That's the mainly three frameworks I found. And the problem for everyone is that this is not a unified, there is no unified framework that can you use like a database that stores every information you can have for predict, a database that stores the interaction records you have. And there's no unified data warehouse where you grab the information and just model. Also, most of the models in machine learning are black box models. So that's a problem when you want to interpret what's happened. They tend to predict good, but it's hard to explain. And of course, if you don't have all the infrastructure here, it's very possible to have a cloud service or an app or something that make the prediction useful for the people that are not scientists. So that's very difficult, but that's the landscape now. There is no system now that implements these things and can predict the new interactions in the future. Okay, so what we want to do is to tackle some of these problems that is not completely finished. I'm trying to go in this direction, but the main thing is to create a big that data warehouse where to store every information that we can put the machine learning model and simplify the models with some math to decide to train with lower information amount to be faster, and then try to compare different systems to see if our system is really predicting interactions, not in the basis of some case specific. I mean, we want to compare some kind of interaction like virus and bats is similar to other interaction with grasshoppers and plants or something like that are parasites, but not the same kind of parasite. Okay, so I call this machine learning framework Ecointeraction. So what we want to measure is there are two things. One is this susceptibility. This is the concept I'm using and adapting to this machine learning framework. That is how a host species could be susceptible to a parasite given the information I have between all the other hosts. And how can I compare in different taxonomical units, for example, a full genus or family. And this ranks from 0 to 1. So I can interpret the outputs from the machine learning at this susceptibility, because the outputs from machine learning in the classification are from 0 to 1. And that's something that I'm using to cut off the data I have. The fact that the information relates in the interactions counts you have in a database relates in this power law distribution. So the good you feed the power law distribution in the data is the good of the data, the quality of the data you have. And we can keep the most of the data with the lowest number of hosts. That means these hosts, this 20% of hosts, holds 80% of the information. So if I train my models with lower information, it will be faster, but I can predict everything in the system. So this thing do something similar and use some ideas similar, and they give. I published this before them, but they claim this before me, so it's fine. But So this is our methodology, the big picture of the methodology. We want, we select some hosts that we know that are related with some specific parasites. And then we use very specific data between the hosts. In this case, we use the phylogenetic signal between the hosts, that is the phylogenetic distance between two hosts, but also the environmental distance between the known environments of the host and the geographical overlapping of two hosts. So with these pairs, we can match if a host share, well, suppose you have two hosts, host A and host B. If they share the same parasite, they should be closed in this data set I'm providing. So we get the pathogens that are related with this host and label our system with the pathogens we know, and the pathogens we don't observe, we label as unknown. We prepare the data and put in the machine. For this specific problem, I use an algorithm called random forest, but you can use more than that. And at the end, you get a susceptibility estimation, given the machine learning, that host could potentially be on risk for a particular system, like hantavirus or other system, for example, for rodents, or COVID for bats, or any system like that. Then we analyze the results and see geographical patterns, and also put the susceptibility on the phylogenetic tree as a trait to see what happens there. But in the very specific case of study I shown here, I focus on rodents and I do two steps, a global prediction of all the known potential hosts and the unknown potential hosts. That's it, try to find new hosts of in this case, I use hantavirus because I want to see which rodents are more susceptible to any kind of hantavirus in the databases. And the idea is to predict new interactions or new hosts that has not been recorded as hosts of a specific hantavirus. So I do this for all the world, and I do for endemic virus, finding a specific interaction of a specific virus. The first one, the global model, is not specific, it's just at the level of genomes. And the local study here in the endemic province of North America tries to find new interactions. So yeah, what we want to do in the global assessment is find the rodents that are susceptible to all the family of Cantaviridae. In the local assessment, we want to find new interactions. That's more powerful because there are some interactions that has not been recorded, and our model provides some information there. And then try to see what happened to the power law distribution. I put this at the end because it's important, but it's not the main thing here. Okay. So the global assessment takes around 1,700 species of rodents. Then I collapse all the information and in a risk map and see the patterns. And the local model is around 170 species. Then I find the specific virus that are from the Cantabria family. Or no, I think I just take any virus. Yeah, in the local system, I take any virus in the global database. Yeah, because I want to find new interactions, not specifically hantavirus. And then I perform my models, and then I perform a 5-parameter fine tuning to have the best model in each run, replicate a lot of times, and then predict new links. That means I have a problem and have never seen one virus, but my model says that they potentially could be a valid interaction. And then I try to validate it with the literature if somebody find that, but it's outside the database. Okay, so these are the results of the global assessment of the potential host of the hantavirus and where are located. These are the counts of the species. This is the richness of species that are susceptible of hantavirus. And you can see some regions that are important and could be potentially dangerous for this. And these are our metrics of the estimation of the metrics of the accuracy of our models. So you can see we have fair metrics. And these are for the findings of new interactions. So we have, we found 652 novel interactions because we only have record of 45 interactions in the endemic rodents in North America, and the machine found 10, well, like 10 more times, like 15 more times interactions that actually we trained with only 46. And we support these replicating until we have certainty that it works. Okay, and we use these three metrics. This PPD is that we can catch interactions that we separate in another database that are valid because we only have positives there. I mean, there are interactions that we know happens, but we don't put in the model, and we validate with this database, and it can catch highly these ones. So these are some of the results. These are hosts that are in the original database. That means that we have very low number of counts. I mean, this work, we expand this with new findings. We multiply the knowledge we have. And then we go to the papers to see if this interaction actually has been reported, but not recorded in the database, because this is very common. Somebody can report this, but the curators of the database doesn't have a way to automatically push this on the database. So I found some of this interaction happening with this paper, and I should be more rigorous looking this. But yeah, I found some host that has recorded this virus at my model, my machine learning model found, but has not been in the database. So that's promising because support the model behind, and that's it. So this is the summary of what I done. I tried to automate the data collection and the retrieving of papers and the integration of the literature. Also, I tried to support the understanding of the geographical distribution of not the scale, the biological interaction, but more the genetic structure of the population, and potentially add the interactions there to see what's happened with that. And the third was to build up these frameworks to expand our knowledge with the basis, but trying to provide predictive results that can impact faster than what we have now. So thank you, and I wait for your questions.
I have one about the second effort, and I don't know enough about niche modeling, but I understand how you calculate the SPA, but that relies on environmental suitability that you estimate somehow. So how do you, is it just where they are right now?
No, I mean, there are many assumptions there. Yeah, you take the observations of the species now.
Right.
Yeah, and then you go, actually, Dan and me are doing this species distribution modeling.
Right.
But the point of view I'm using here is we can call that the old school of the species distribution model, that is Jorge and Tam's school,
right.
You know, here. What they say, and basically, it's that there is something that we call the environmental niche. That's it. If you go outside these environmental values, your species cannot grow. So your species will be confined in environmental space and a surface of variables. In this case, you can imagine precipitation and temperature. And what you are doing is just drawing a surface of combinations of precipitation and temperature, that you're saying and assuming that if your species are outside that, your species cannot grow. So what you do is take a random sampling of points everywhere that you suppose are absent and compare how is the environment there. And you have the locations of the species now, and you use that information to provide to another machine learning algorithm to classify if a location with some environmental combinations could be or not could be suitable for the species. So you draw like a layer.
Yeah, I get to SDM. I guess what I'm saying is that your environmental variables are dynamic right now and over 820,000 years. And your species are dynamic, and yet you only have, they're here now.
Yeah.
And here's the environment in which they like now.
So,
right, but over 120,000 years, you could have different preferences for different
no,
environments.
that's the main thing here. I don't put that slide, but I think I should here. There's something that Tan and Jorge call the fundamental niche. That thing is very strange because it's funny. The fundamental niche is beside that. It covers that preference, and it's a surface broader that gives you an estimate that the species can be there even if now or the past of the future the environment change. That is that you take, this enough broad to cover the actual distribution, the potential distribution, and that's the fundamental niche. Because you don't know it a priori, just suppose it, and we do a relaxed version of ecological niche model that is not overfitted. That means that I am not modeling the species preference of the environment now. I am modeling a broader niche that can hold that along the environmental change. That's the key question.
But you assume that that doesn't change through the 120.
That's it. That's why I'm with them. Because we know that the variability is important. And in a new version of this point of view, if you take in account that, that's better approach that you suppose that doesn't, this is fixed and doesn't change, and the movements here are just related with the environment. So this is the dynamics you say. In time one, the geography is static here. The point one is here all the time. You go there, but the environment is not the same all the time. So this surface, you suppose, is static in such way that can evolve, but very slow regarding with the structure of the population genetics in that species. So the fundamental niche evolves, but very slow. That's how Stanford said. That's it. But we know that that's not completely the full story. What we want is taking account this variability. So yeah, we are developing the second stage of this thing with them that takes in account that. Because if you observe the variability and you model it, and then you have the variability on the past, this will be better and better and better. But yeah, we are under that assumption. That's it. Okay.
So on your third section, did you do any leave and out validation of the stability and sensitivity of your predictions? And if so, what did you find in terms of substructure influence of what kind of data you have access to? I know that a lot of these host-parasite interactive networks have a lot of biases in terms of where we've collected data, what we have evidence of. So being able to extend that would be pretty reliant on getting a sense of how stability of your estimates rely on your input data.
In the first part the trick was here. This part proportion of the species has more of the counts. So when you observe these and you remove in your modeling process with cross-validation and you remove information and feed, these catch that because suppose, for example, in this case, you have bunch of information for one species, and if you remove their information, it fits in. It doesn't matter if you remove some of the interactions or here, because you are using just this portion. So yeah, I do some I do cross validation all the models and replicate the cross validation. So I do 10, 10. Yeah, that's it. I do 10 replications of the cross validation, and the cross validation, I remove 10 and model and repeat, and remove 10 and model and repeat, and do that 1,000 times. So with that, I can ensure that each run of these has a statistical distribution of the susceptibility, and that is how I catch the error. That is how we do.
So I guess the question for me is, if we want to apply this framework to less studied systems, and we want to extend this out to them, how it kind of amounts to what proportion of that 10 or 20% that have the most information can you be missing and still get stable estimates? That's kind of my question, is how much can we understudy the most important taxa in our model before we start to get failure states?
Yeah, I didn't measure that, and that's true. I suppose that if I remove completely one, I should be able to predict that one with the others.
Okay, but it's generally just very dependent on that we've
Yeah,
done proper
but
rich
the
sampling of that most important.
Uncertainty tends to gets worse when you have less information. That's it. It's what you expect. So there's a threshold where you put the information and say that everything over that it's valid, but yeah, tends to get biased to the species that has low observations in the database. But now I have a better understanding how to measure that. And I found this property of the power law distribution very common in all the databases. And I can use that as a measure of the quality of the database. Because, for example, this case is very clear. This database has been curated by hand with experts in a specific paper that has counts of birds and West Nile virus that are detected in mosquitoes that infect these birds. So this curated by hand by experts fits better than these that I gave there everywhere in papers that are on the level of family, each coronavirus there. So you can see the fit is not good. So the goodness of fit in this power law distribution gives you the quality of the data. And then when you predict here, you can expect until here, this point, that is, you can have a fair fitting. But here, these two, probably not. That's how it works. This is my measure of quality. But I actually don't do exactly research how this can be affected and which is the state of the actual databases in the quality of the data. But yeah, because that means that I need to curate by hand that, and I don't have time to do that, but at some point, potentially get some collaborations there. And if you curate by hand and you know that these counts are good, it fits better. That's it. It's the only way you can ensure that error doesn't propagate in the low observation part of the data. That's it.
So back to the Sky Island
Yeah.
one. I forget which one that was, number 2, I guess.
Yeah.
Do you have a sense of how many different species there might be where you could basically do the same kind of study that are, confined to Sky Islands like that, maybe even in this same part of the world?
Now I have a better idea because a new database has been released a few months ago that it calls Gen Deep or something like that. It's a genetic diversity database that gather papers with this information, not exactly fixation index, but other genetic diversity index, and are georeferenced with the GBIF dataset. So you have GBIF dataset, as I do, but reference with one by one reference to the genetic diversity for that population. And they ensure curating, this team ensure that you have enough information along all the distribution of the species. Suppose you have a species that lives here in the Sky Island, so you just filter the species that lives here and that you can gather from that database, and you will ensure that you have enough information along here. They neglect everything that could have one or two populations study. They try to cover all the range of the species. So if I match that new database, yes, I can potentially know how many species living here has similar information in the genetic structure and replicate this massively. That's my next step. I'm dealing with that, but yeah, they have this new database and it looks good. They are from Switzerland and they invest a lot of money in that database.
So we reached an hour, so I guess people are allowed to slip out now with that.
Well, thank
Sorry,
you.
you have another thing. Thanks again.
Thank you.